start?
Okay, well, so while that's loading, today I want to talk to you a little bit about some work I've been doing on some data sets that I picked up when I joined Tom Younger's lab here at University of Texas that I think collectively aim to study floral divergence patterns in a subspecies pair of Ipomopsis. And so Ipomopsis is a reasonably speciose genus and that expresses remarkable floral diversity both across species as well as within species. Much of this is thought to be driven by pollinator-mediated selection and pollinator specialists and so forth. And so really it's been a really key model in understanding floral evolution for the past several decades. So today I want to talk to you about a subspecies pair, the Ipomopsis aggregata species complex. So on the left here we have subspecies Candida, which produces these elongated white flowers that are often observed to be visited by hawk moths in the field. And then on the right here we have Ipomopsis aggregata Kalina, which produces these shorter red flowers, which are often observed to be visited by hummingbirds. Now these two subspecies do occupy slightly different portions of the overall aggregata range, but they do overlap in the front range of the Colorado Rockies, where they do hybridize in nature. There's a pretty rich history led by the late Vern Grant that has really studied speciation within aggregata, which I'm not going to get into today. Instead, I'm going to focus on sort of the more contemporary roles of evolution in maintaining subspecies diversity within this region. So recent work that was actually very recently accepted in molecular ecology that's coming out of Tom's lab, carried out by a couple of former graduate students, looked at some ecological genetics across the Front Range. And so what they did is measured a bunch of phenotypes, floral phenotypes across the Front Range, and then compared this to a set of neutrally evolving microsat loci. And what they found is that several traits exhibited elevated quantitative genetic divergence relative to neutral expectations. So normally we take this as evidence that divergent natural selection is acting on these traits or has acted on these traits. And this low overall global FST value suggests that there's a considerable amount of gene flow that's potentially occurring across these populations. And so with that, my main questions with these data sets were what are the major drivers of selection acting on floral divergence in this hybrid zone? For this, I'll present a very brief selection analysis. Next, what is the genetic architecture of some of these traits that are showing floral divergence? And for that, I'll be presenting a QTL mapping approach. And then finally, how representative are these results across the entire Front Range? And here I'm going to talk about a candidate gene that we think is potentially involved in floral diversity in this subspecies pair. I should mention at this point, as I said, I inherited this data set from these folks up here, Elizabeth Milano, Amanda Kenny, and Jacob Sewell, all former graduate students of Tom's. And then, of course, Robin Hopkins, who was a postdoc at UT, now faculty at Harvard. So they employed an experimental population that was derived from a 4-way outbred cross. So 4 grandparents from 4 different populations were split off into a white and red pair, crossed to generate an F1, which was reciprocally crossed to generate a large outbred F2 population consisting of nearly 800 individuals. And importantly, these guys segregate full spectrum of flower color as well as many other quantitative traits. So they planted these individuals into a field population in the Front Range hybrid zone. They measured a whole bunch of traits, including flowering time, a bunch of traits which I'm going to collapse using a principal component analysis into an overall size descriptor. So what does the pollinator see when it enters the field? They also measured 7 morphological features of flowers. Again, I'm going to reduce the dimensionality using a principal component analysis into a length and width descriptor for flowers. They scored color, nectar volume, they also followed pollinators in the field. So we have some good data on visitation rates from our 2 primary pollinators. And then finally, harvested fruit at the end of the season, which I will use as an estimate of lifetime fitness. Okay, so this is just a mock diagram. This is how I analyzed my data. I used a path analysis approach. So here, all I'm asking is, what is the direct effect of all of these phenotypes, as well as visitation, on overall fruit production, while considering correlations between these traits? And then I asked, what are the effect of these phenotypes on visitation rates? Okay, so here's what our data look like. The first thing to note is that there is a direct relationship between flowering time and fruit production, and it's a hashed line indicating a negative relationship. So earlier flowering plants produce more fruits. That's a pretty typical result that we see in plant selection work. Also, plant size, width, and nectar volume all have direct positive effects on fitness. And of course, not surprisingly, both pollinators also positively influence fitness. If we step back and ask what these traits are, how these traits are affecting visitation, what we can see is that hummingbirds search out and find larger plants in this experimental population. Both hummingbirds and hawk moths like a lot of nectar. That's probably not surprising. They have the capacity to learn where the reward is. And we were able to confirm this divergent sort of pollinator preference on flower color, where hummingbirds were actively choosing red flowers, whereas hawk moths were actively selecting white or pale pink flowers. Okay, so with that, the next question we really had is, what is sort of the underlying genetic architecture of some of these traits? As I said, I'll be using a QTL mapping approach. So here, I attempted to map all 9, this laser pointer is just running out. So I attempted to map all 9 traits that I presented in the previous analysis. And I'm just going to quickly show you the overall kind of result. I don't want you to get bogged down in the details here. We were able to, so first of all, we recovered 7 linkage groups, which is consistent with the number of chromosomes in the system. So these lines here represent our linkage groups with marker positions along them. And we were able to map 17 QTL across 7 of the 9 traits. This included a couple of QTL for hummingbird visitation. So while that's not a phenotype, we still have the ability to map it, as well as a couple of fitness loci. And so one thing I want to point out here is that there's many potentially overlapping QTL. I guess I should say QTL are these colored bars, where the length of the colored bar represents the confidence interval around the position of that QTL. So this suggests that there's potentially quite a bit of pleiotropy or tight linkage happening between QTL. Now, I want to go over a couple of these QTL in a little bit more detail. So the first couple of things that I'm pointing out here is that Collina alleles, these are alleles that were descended from red grandparents, not surprisingly make red flowers, but they also lead to greater nectar volume. So these are just raw effect plots taken from our QTL analysis. Along the x-axis here, Candida refers to Candida homozygotes. Collina refers to Collina homozygotes, and then we have 2 heterozygotes. And yeah, so what we can see is we got white flowers for Candida, red for Collina, with some intermediate for the heterozygotes. And that Collina alleles tended to produce greater nectar volume. So if pollinators are showing preferences for flower color in this subspecies pair, this could lead to divergence in nectar volume. Next, I'm showing you here the same effect plots, but for PC length and PC, so this is flower length and flower width. And what we can see is that these alleles descended from red grandparents mostly lead to shorter and wider flowers. So they lead to shorter flowers for that length QTL and wider flowers for this width QTL. Now I say mostly because this pattern is upheld for all our QTL that we discovered for length, but only in 2 of 4 width QTL do we actually get sort of the pattern that we expect. At the other 2, we get Candida alleles also producing slightly wider flowers. So there's something interesting going on there that I'm not entirely certain how to explain. Okay, so we find evidence that alleles descended from different grandparents lead to traits that we observe divergence on. The last QTL I want to talk about is this flower color QTL we discovered on linkage group 1 in our population. And the reason why this QTL is pretty interesting is it was the largest effect QTL of all 7 QTL that we found. It explains greater than 30% of flower color variation in our experimental population, so that's a pretty large amount. And also, it localizes to an express sequence tag for a gene called dihydroflavanol 4 reductase, which some of you probably know as DFR, which has been implicated in color transitions in several other species. So we think we have a pretty good flower color candidate for floral diversity in this subspecies. But the question really is, how representative is this? We only sampled 4 grandparents, so it's possible that we just got lucky to sample one with a polymorphism in this particular gene. So to answer that question, they revisited populations in the Front Range, harvested flower tissue, first scored flower color, and then harvested flower tissue and scored gene expression at DFR. And if we look at just a straight correlation between these, we see a pretty striking result in that high DFR expression leads to red flowers. And so the way that we measure DFR expression means that lower numbers correspond to higher expression, and lower values of our color metric also mean red flowers, so we see there's an association between expression and flower color. And this pattern is upheld if we look at the mean values for populations as well. Okay, so just to sort of wrap things up, so what are the major drivers of selection that we determined in the field? We found that large, earlier flowering plants that produce wide flowers with a lot of nectar tended to be more fit in that field population, and that pollinators do exhibit preferences for color only in this experiment, but not necessarily other traits. What is the overall genetic architecture? We find a lot of QTL for our traits, several of them potentially overlapping. We were able to confirm that Kalina and Candida alleles differ for several of the other traits that show divergence, including volume, flower size, and shape. But interestingly, we don't necessarily see, or we didn't see in our experiment, divergent preferences on some of those traits. So perhaps there's some undetected pollinator-mediated selection occurring. And then finally, we seem to find evidence that DFR may be a recurrent target for pollinator-mediated selection across the Front Range. Of course, we'd have to follow up with cloning and transgenics and so forth to really confirm the function of DFR in floral diversity, but we at least have pretty good evidence at this point. Okay, so with that, I'd like to pay special thanks to John Lovell, a postdoc in our lab, for help with QTL mapping, the younger lab, and thank you guys. Time for a question.
So hummingbird visitation QTL that co-located with what? I noticed that there was...
It co-locates with width, color, and I think another one co-locates with volume.
But you didn't find any moth visitation QTL?
No, yeah, so in that season, and I think this is why we didn't detect it, in that season, they got an abundance of hummingbird visitation relative to hawk moths, so just might not be enough data there to show up.